Good morning, everybody. Good morning. Happy Friday. So this morning, as you know, we have the. We lose the slide. I think was there a. Okay, no worries. But this morning, as you know, we have a seminar entitled. Well, it's a group of talks, I would say, rather than a seminar, and more of a symposium, if you like, entitled "The Path to Commercializing Gene-Edited Plants and Microbes," and we have some really distinguished folks from inside the university and outside the university who are going to talk to this subject. And then at the end, we hope we're going to get a good Q&A going. I'm really delighted to see so many folks from the university, but also many folks from outside the university. So it's great that you've got word of this, and you're really welcome always to attend these events and seminars and symposiums that we're holding here at the Plant Sciences Initiative. We really want this to be a convening center, and so you're very welcome. And it's great to see old friends and old colleagues as well from my previous lives in the industry. So my name is Adrian Percy. I'm the executive director here. For those of you who are not aware of the Plant Sciences Initiative, we're actually fairly new. You can still kind of smell the new car feeling in this building. We only opened the building last April, so or April before last now, so 18 months ago. And we are now the home of round about 25, 26 interdisciplinary programs who are all working on really critical grand challenges in agriculture and environmental health. So we boast scientists who are coming from all over the university, whether it's from the College of Ag and Life Sciences, from the College of Engineering, the College of Natural Resources, and others. As I said, and they're working in interdisciplinary teams. We're actually bigger than that, though, because across campus now we have 50 to 60 affiliated faculty. And again, what we're trying to do with these faculty who are coming from very, very different backgrounds is get them to convene in this building, and again, to form teams and to work collaboratively around some of these grand challenges. Outside of that, we're trying to make impact through commercialization, and that can take several routes. One, of course, is to work with industry partners, and you're going to hear from some of them today. And we have some great partners, companies like Bayer, BSF, Novozymes, I should say, not Novo Nordisk, SAS, and others, some of who have taken space in this building. And we work through them through our Office of Research Commercialization, basically tech transfer, folks like Coltarin. But then we also have a big entrepreneurship effort. And we're really proud now that we actually have 7 companies who are being spun out of the science here. So that's a way we can make sure that our science is relevant and hopefully gets scaled up at the field and serves our constituents, which are mainly growers in this state and other states across the US. So that's a little kind of overview for those of you who don't know the PSI. But today, this seminar really intersects several areas that we're very interested in. One, of course, is commercialization, as I've just said. So either on the entrepreneurship side or just on IP generation. And that's one of the reasons that we wanted to have this seminar, to bring together, for instance, researchers from the university who are working on topics which may have relevance in the area of gene editing, like Jack Wang, who's going to talk in a second. And basically advise them and get your collective wisdom, hopefully, over the course of this event. Secondly, one of the programs that we host is called CIRSA. And Danesha, I hope I get this right, the Center for Excellence in Regulatory Science in Agriculture. It's a bit of a mouthful, but once you come up, so Danesha Seth-Kali is the director of that center. And since we are kind of jointly hosting this event, I just wanted to invite Danesha to give you the kind of lowdown on CIRSA for a couple of seconds.
Okay. Good morning, everybody. It's so nice to see you. I know about half of you. So good to see you, Alan. And those of you I don't know, you'll feel like you know me after I'm done talking. And I have to say, anyone who already knows me knows that there's no way I can give a 30-minute synopsis. So I wrote it down. Otherwise, I'd be up here for 45 minutes and I would take your time up. So CIRSA is the Center of Excellence for Regulatory Science in Agriculture, as Adrian correctly named it. We call it CIRSA for obvious reasons. So that's maybe when you hear it, that's what we're talking about. And we were sort of the flagship group under PSI even before Adrian's time. So we're really proud that we've been operating even longer than the building has been here and even a little bit before most of the programs here gelled. But we feel like we sort of have a toe or a finger hold in each of those programs. So in short, and again, this is why I wrote it down, otherwise I'd be up here for 45 minutes. CIRSA is an initiative that tackles challenges associated with advancing knowledge around regulatory science, especially as it pertains to agriculture and in the agricultural arena. We work with federal and state agencies, especially, of course, the regulating agencies, but we work with other ones as well. And we work with a number of industry partners, academics, of course, and NGOs. Our goal is to advance transparency and understanding and open a dialogue around regulatory policy as it relates to agriculture. So that's us in a nutshell. So I'm more than happy. I'll be here all morning. I'm more than happy to talk more about our initiative. We have a poor website that I'm still working on creating and adding content to, but any of the PSI folks in the room can tell you more about our program. And we even have a few board members. I'm just going to call them out. So Randy, Laura, Dan, you guys want to just raise your hand. So we have a few board members in the room. Feel free to grab them and talk with them as well. Thank you so much, Adrian, and I will turn the floor to you.
So without further ado, we're going to invite our first speaker, Professor Jack Wang, to come and talk. And if I can just prelude it, no, come up, Jack. I'm not going to give you your bio because you can talk to them about yourself. But we're really proud to have part of Jack's program in this building. In fact, if you go upstairs to our greenhouse on the roof, there are some gene-edited poplar and eucalyptus trees, and we have an association, shall we say, with Trico, which is the company that Jack has founded. But please, Jack, we welcome your remarks.
Thank you, Adrian, and thanks for the opportunity to be here. It's great to see all of you this morning, this very chilly morning. So I'm here for quick 5 minutes just to really tell you a story about trees and the exploration of genetic engineering trees and the historical perspectives of many of the important things that we can learn from. And this is, much of it is based off personal experience, others based on the experience of my colleagues. So, and henceforth, the title of this talk, where, there's a lot that we can learn from the past in order to understand the future. So as academic scientists, as researchers, much of our work tailors towards the first two quadrants, science and technology, exciting projects, exciting discoveries. Occasionally, we venture into turning those technologies and sciences into applications. And then, if we're lucky, we get to produce some very cool products. Drought-tolerant plants, disease-resistant varieties, fast growth, all of those things. And then we pat ourselves on the back, job all done, on to the next project. But often than not, what we missed out is the most important part of deploying the technology, and that is acceptance. Acceptance by governmental agencies, acceptance by regulatory framework, acceptance by society, the general public. That is often the most important and the biggest barrier to successful technology deployment. So I'm just going to tell a quick story about trees. I'm guessing not many of you worked on trees, so this might be a new story to you, and it's a pretty good one. That beautiful place in the backdrop, that's New Zealand. It's my hometown where I grew up and where I lived for many, many years before coming to the great US, and especially this fantastic university. And it's actually Hobbit Town, where the hobbits lived. It's about a 30-minute drive from my house in New Zealand. And there's a very good reason why I'm here in the US and not back home living amongst the hobbits. And that's because New Zealand, as of right now, is one of very few countries in the world that's completely GMO free. In fact, you can't even go to the grocery store and buy a papaya because, of course, that's a GMO. So as you can imagine, being a biotechnologist in a GMO-free country, my career outlook wasn't very positive, and that's why I came to this fantastic country. But New Zealand wasn't always like this. In fact, back in the late 80s, early 90s, if you consider outside of the US, New Zealand was considered one of the R&D powerhouses of the world, with some of the most innovative technologies, know-how discoveries surrounding biotechnology with technologies that are even used today in forestry, in fact, in terms of delivering regeneration of transgenesis, and they produce some of the most fantastic GMO plants and trees ever. So why did we go from that to where we are today? Well, as it turned out, it was that last part, the acceptance part, that went wrong. So I could speak about this for hours and hours and hours and bore you to no end, but I'll try to condense it. Essentially, what happened was the academians, the industries, and the governmental agencies during the golden ages of biotech were overly enthusiastic about the prospects of GMOs, and they really oversell the technology back in those early days, focus on the benefits, focus on the values, and downplay to a large part of the potential risks and challenges of these. And so all you see are, GMO is the savior of agriculture, GMO is the savior of livestock, GMO is the savior of forests. And eventually, during those early years, of course, a lot of experience got wrong, a lot of field trials got wrong. We have all sorts of abnormal developmental traits that occurred, and those are all brushed away, downplayed, or hidden in a closet somewhere. But the public eventually found out about those, and so it brewed tension, it brews distrust, it brews from the lack of transparency. NGOs that eventually just took things into their own hands. So there was a momentous event or a pivotal event in history where it changed everything, and that was Christian Walter, considered a leader in our field in forest biotechnology, who created some of the most amazing first generation GMO trees in New Zealand. He went through all the processes, his career's worth millions of dollars, and even governmental deregulation for field trials of around 400 transgenic radiata pine, the most important economic tree species in the Pacific. And so he planted it. He actually found a secret location in the middle of a forest. There were 2 layers of electric fences. There were armed guards and surveillance. But what the NGOs did was, in the middle of the night, on a holiday, they dug underground tunnels underneath electric fences. A group of them went in with chainsaws and cut down all of those trees. In fact, they went beyond that. They actually plugged up all the stumps and cut the roots off. There was no way to recover those. Beyond devastation or financial losses, the loss of years of research and all those efforts spent, the devastation of the morale of scientists was what really hit home. So the scientists, the biotechnologists, the geneticists of New Zealand, after such an event, generally they retired, or they moved on to something else, traditional breeding, for example, or they came to this fantastic country. Long story short, the NGOs has grew since then. In fact, the same thing happened in the US, in Oregon. There's a field trial that was cut down in South America, in Europe, to a stage where conventional GMOs nowadays in forestry just doesn't exist. In fact, there's not a single GMO plantation forest in the world. Now, a silver lining to all of this is we got a second chance. It's very rare in life that we get a second chance, but CRISPR offers that to us because the technology is fundamentally different to that of a conventional first generation GMO, where we have the ability now to harness a tree or cell, a plant's natural DNA repair mechanism in ways that have never been done before, in ways that create novel genetic variants that are genetically indistinguishable from natural variants. A powerful technology. So with this challenge, we really have to be mindful now to separate gene editing from conventional genetic engineering, and then highlight the values of this technology, but at the same time be transparent about the potential risks and the considerations that are needed to make sure that this technology is deployed in a way that is safe, that is rigorous, and that is done with environmental assessment and considerations in place. So with that, I think there's a very bright future. If we can debottleneck the acceptance part of the technology deployment and get a public endorsement, I think there's an amazing future for biotech plants and CRISPR plans to address some of our grandest and most critical problems in society today. Thank you.
Thanks, Jack, for giving us some context and some history. I have to say, one of my best friends, his farm backs up to Hobbiton.
Oh.
And so we need to talk because you probably know each other or know his family perhaps. So it would be great. So I think we're going to move now on to our industry speakers. So we have 3 industry speakers that are going to give their perspectives on microbes and then plants and then plants. First of all, we have Natasha. Natasha, thank you so much for giving up your time. Natasha's with Bayer Crop Science, of course, and she's been involved in a lot of the events that we've already held here and a very active member of our community. So please give a welcome to Natasha Dixon.
Good morning, everyone. Again, thank you, North Carolina State, for having Bayer at this event. We really appreciate the opportunity to speak to you for a brief time today about the commercialization of gene-edited microbials in plants. So our quick agenda, we're going to just a quick introduction to Bayer. We're going to talk about ag biological products, and then we are going to finish our talk with gene-edited plants. So just a brief introduction. Bayer really believes in the mission of health for all, hunger for none. And our colleagues here in the RTP area, we try to live by that motto as we do our work. We are in the crop science division, and innovation is a really integral part for Bayer, right? Either between our collaborations with outside, our work inside, we really believe that innovation will fuel the future and then provide the mission that I just mentioned, that we will have health for all and hunger for none. So again, I mentioned we're in the crop science division. We have about 34,000 employees. We have about a $25.2 billion worth of sales, a $2.6 billion research budget, and we're active in over 143 countries worldwide. So I want to start off this talk. Really, I divided my talk into sections. So biological products, why now, right? Anytime I talk about biological products, I really like to show this slide to show the diversity of what we consider biological products. So you can have anywhere from a biofertilizer that could be a microbial, you can have biostimulant type products, and then you move over to your biocontrol side, and then you can have biopesticides, and then under that you can have biochemicals or microbials, and then as you can see the different division and sections underneath there. So one of the things also that is really important about this slide is that the mode of action is really important when developing your regulatory pathway. So even though maybe on the biofertilizer and the biostimulant side looks similar, you may have similar actives using both in a fertilizer way or a biostimulant way. You really have to understand, and when you're developing your project, the claims, intent, and mode of action so that you can contact the appropriate regulatory agency. So biological products has been around for a while, right? This isn't anything new. We've had BT, we've had even products in Bayer that's been on the market for over 20 years. So why the resurgence now? I like to call it the biological products revolution, right? So why now? So I would like to give you 3 factors to consider on why the products now are being considered more important and being used in a integrated pest management solution or tailored solutions. But I do want to caution, like Dr. Wang did in the beginning, we don't want to overpromise and under-deliver, right? And I think that was one of the issues when biological products first came on the market. We thought that we would be able to do all these different things. And at that time, maybe clarity, a mode of action, what we were doing, what we were using wasn't clear. And then farmers got the reputation of biological products being snake oil, right? And it didn't work, and it didn't work for my soil, so therefore it doesn't work in general. So I think the new variety and new generation of biological products are a lot better in addressing the needs and also understanding what the products can and cannot do and giving that message back to the farmers. So the first thing is environmental considerations. So climate change is here and it's here to stay. And unfortunately, it impacts farmers in a way where you have extreme weather events and your soil may not be healthy, and therefore unhealthy soil equals unhealthy plants. So biological products could potentially help with climate change mitigations by providing a healthy soil for a healthy plant, or a healthy plant to help withstand drought tolerance or over watering or heat extremes, etc. The second thing under environmental considerations is actually the farmers limiting their environmental impact. Using biologicals in a responsible tailored solution program where you may spray with chemistry, go back under biologics. You may not have to spray as much, therefore the farmer's impact may be limited on how much gas he uses to go in the field, how much effort it takes to actually harvest with just a pure chemical footprint. Secondly, which is really important, consumer considerations, right? So we know that sustainability is not just a buzzword, it's here to stay. And so consumers want to know that their food is being harvested and being farmed sustainably, and that could mean less chemistries in some cases. It could mean just the way that again, healthy soil, happy plants, right? So they're really wanting to know that their food has come from a sustainable source. And then also food security. If anything, COVID taught us that supply chain issues can really be a problem, right? And so if you have a biological product that can help with yield, that way you can have food all year round. We like our strawberries in June, and they may not be grown in June, or our avocados all year round. You know, with a yield increase that could be a potential for these type products, then you can have your food security, your local food security, and your produce all year around. And finally, the part that Lieselot and I sometimes lose sleep over is the regulatory considerations, right? So we know that in parts of the world that chemistry use is being reduced or restricted. We know its ambition goals in our friendly country, the EU. They want to reduce pesticide use by 50%. Therefore, you need products for the farmers to have. And again, using these products responsibly, I think that's the key word, responsibly, with an integrated pest management system, meaning again, chemistry and a biologic, or however your farm plan may work, I think is a solution to be able to provide farmers globally with products when they are losing some chemistries. Also, again, going back to that, you know, with the reduction of chemistries in certain parts of the world, MRLs are really important for the farmers to even be able to import their hard work into another country. So having a biological at the end of a spray program will help you reduce your MRL considerations during import. So where does Bayer fit into this? We really believe in the power of biological products. We believe it can go into regenerative ag, meaning doing more with what you have. We're not getting any more land. We want healthy soils, healthy plants. We want responsible farming. And so Bayer really has an open innovation model that really fosters, hopefully bringing these products to market responsibly, safely, and quickly. So we have a couple of open partnerships. We only mentioned a couple on the slide. We have a really big partnership with Ginkgo Bioworks. We have a partnership with Chemitech. We have an arm of Bayer called Leaps by Bayer, who also funds academics. And then out of that, we also have collaborations from Andes, Sound, and Pivot Bio. So those are just some of the highlights in our open innovation model. So now getting to the regulatory nitty gritty, right? So you just heard me say, oh my gosh, these products can really work. Like we've evolve them from first generation products. You know, they can help with climate change and they can do all this different stuff responsibly and on target, not over promising and under delivering. So why aren't biological products flooding the market right about now, right? Why aren't you seeing more farmers adopt biological products? And the answer to that question is it's actually complicated, right? So for regular biocontrol registration, not even GE biocontrol regulations, is very hard. So for instance, in the United States, we do have a well-organized regulatory pathway for these products. We have a separate division within EPA that reviews these products. We have clearly defined regulatory requirements. We have divided them into 3 categories here in the United States. And then we have established timelines, meaning that we know when we put a product in due to PREA, we will get a product out in the appropriate time. I know some regulatory people in the audience are laughing about that. And I'm sure they can talk to you afterwards, but we're going to be positive today. And our average time of a biocontrol product here in the United States is 18 months for a new product. So our EU folks, they are a little bit different. I mean, we have made strides in the EU. There are separate biological testing requirements that just got approved last year to be in effect this year. So that's a good thing. However, biological products, biocontrol products in the EU are still under their chemical guidelines. So previously, before the new testing guidelines came out, we were doing a mesh between chemical guidelines and other OECD guidelines, which could be harmonized for biological testing, but it was a mishmash. So Europe realized that with their ambitious goals for the Green Deal and Farm to Fork, they need to do a little bit of clarification on testing guidelines. So I'm going to take a step back because regardless of all the regions, what we really strive to have is fit for purpose regulatory requirements and testing guidances for microbial products. If you are still using chemical guidelines for microbial products, or I'm sorry, biological products, that is not fit for purpose. And a lot of times the testing, we cannot achieve the same testing standards as a synthetic chemistry. The EU timeline is really what's the most difficult thing. Even with the biological product, it is an average between 5 to 7 years to get your product out of the EU for a new biological registration. So LATAM, Brazil is our shining star right now in LATAM, Argentina as well. So Brazil does have defined definitions very similar to the US, very harmonized to the US. Also very much so the testing, the tiered testing approach is very similar to the US. However, other countries in the LATAM region either have no biologics guidances at all, no formal definitions, or they go with the chemical standards for biological products. And again, for fit for purpose, sometimes you cannot reach the chemical standards with biological or even especially live biological products. So that's still a work in progress. And then finally with APAC, it really varies country to country. One of the things about APAC is the import requirements are very difficult. Your microbial has to be native to the area, the region, especially in countries like England and Australia, and New Zealand was mentioned as well. If they are not native to that area, more than likely the import time, the time to import the strain is significantly more than the time to register. And on the other side of that slide, biostimulant, biofertilizer registration is definitely not harmonized as well. Here in the United States, we do not have any federal fertilizer agency or regulations. The USDA has established scopes and times for permitting, especially with the edited microbials, and you have to do a state-by-state registration strategy. The EU recently in 2019 passed a new fertilizer regulation or law. So you have 2 options in the EU where you can either try to get a EU-wide certification for your biostimulant, your biofertilizer products, or you can go the traditional EU route and go state by country to get your registration, which again is laborious in time. LATAM is similar. Some have national fertilizing agencies and regulations. I highlighted Brazil again. Brazil also, once you get your national fertilizer registration, you do need to do a state by state in Brazil registration for your product. And again, APAC did not change from the previous slide. It varies country by country. There are a lot of countries out there that are starting their biostimulant journey, trying to get right fit testing and requirements for biostimulant products. However, the import and the permit requirements are still strenuous. And a quick note about the US as well. Even though I have on this slide biostimulant, biofertilizer, biostimulant is not a recognized definition here in the United States. We are still working on that. There is no federally recognized definition. And in fact, each state may have something different in regarding the definition for biostimulant. So that's an ongoing process that we're trying to work on with our industry and academia folks here in the US. So what about the GE microbe? So you're going to see several maps after my presentation today, and you're going to say, okay, Natasha's map looks like this, and everybody else's map is full of colors, right? So we're the new kids, we're the toddlers of the group, right? So the plants are very much more established than what the work is with GE microbes. So I have areas in green, and then I have like the United States is shaded, Australia is shaded, and then we have the man with his question mark for the rest of the world, right? So the reason why these countries are in green or they're shaded is they have some provisions for edited microbials. And in fact, Canada and Brazil have actual edited microbials in the market. On the market, the United States maybe on the biocontrol side, but not on the biofertilizer, biostimulant side. And we'll go a bit into detail with that. Most of the world right now, we're just trying to get them to understand the regular biocontrol registrations, right? It's a fear, especially with the microbials in general, before you do any edits to it. So getting other regulators globally to really understand these products are different than chemistries and we need to treat them differently, not in a bad way, is where the global trend for these products are. We want to do capacity building, and capacity building, even here in the United States about the edited products, is ongoing and will be a very long haul as we move forward to getting these technologies in the hands of the farmers. So a quick case study right here in the United States in the USDA. So, you know, we're here at NC State and somebody has a really blockbuster product and they looked at it and they think it's the mode of action. They're like, okay, it's not a biocontrol. So we think it's abiotic stress reliever. We think it's going to be a biostimulant. We did some edits. Where can we go from here, right? Unfortunately, right now in the US, not far. So with a non-pesticidal mode of action that's genetically engineered, our USDA BRS provides permitting for importation, interstate movement, and environmental release. Currently in the US, there's no commercial pathway for an edited non-cidal mode of action product, right? So 7 CFR Part 340 was updated in 2020, and it updated for our plant colleagues. There was not provisions for microbials there. USDA recognizes that now that they're getting more and more application for permits for edited microbials, that they do need to do something with Part 340. We're going to see how that's going to work out. And the reason why I put on the other side, there is precedence to have a non-GE fertilizer product commercialized under a different branch of USDA. So I think the industry and our academic partners, we need to figure out, do we want to follow the PPQ model and move from the research permit is a commercial permit? Will that work for our microbes? And then how we can best satisfy any governmental requirements with our industry needs and wants, and the end product being in the farmer's hands. And then finally, under EPA, so it's kind of two things, two branches with the EPA and regarding edited microbes. So it's really critical because you heard me mention that we do not have a federal fertilizer governing body. So if you really want to get snazzy and you do a totally intergeneric microbe and have an end product, which meaning you're having DNA from two different genuses and you have a new microbe, you do have to have testing under EPA, and that's under TSCA. Because again, we don't have fertilizer regulations and standards, so TSCA would definitely need to see a full risk assessment for that new microbe, and then you will be under permitting conditions there. But actually TSCA does have a way for you to commercialize that product. So that's hope for that. And then finally, the most easiest path in the United States currently, even though it is probably will be some challenges later on, is the pesticidal part. We have an emerging technology branch that handles all edited microbials and products. And again, the average time right now is between 18 months. So with that, I want to thank you for your attention and give you the last few key takeaways. So just again, the regulatory part people always regulatory, right? We get the sad face when the regulatory people talk. But what I really want to let you guys know is please continue to innovate. We know the regulations globally have to catch up to your innovations, but if you don't have innovations to present to any other regulatory authorities, then we will not be able to move this forward and get these products, the interesting products that most of you all are developing, into the market. And again, ag biologicals, I'm not here saying that ag biologicals are here to replace chemicals. They're to be used in a responsible program with chemicals. And then, and maybe, you know, as more edited microbials and products come up, they may have the same standards as chemicals, but right now we really want to use them responsibly in a integrated in an integrated solutions or tailored solutions program. And again, regulatory clarity is needed in general for biological products, not just GE products. And then finally, I really believe that ag biologicals will increase the diversities of what farmers can use, and then provide a really important tool for their toolbox and to be used responsibly for sustainable agriculture in the future. So thank you very much for your time and attention. And then I'm going to bring up my colleague Lieselot, and she's going to do her plant portion. Thank you.
Hello, good morning, everybody. Can you hear me okay? So I'm Lieselot Bertho. I'm in the same team as Natasha. I work on policy topics for the plant side, so GM, as well as genome editing and Bayer crop science. And I'm actually based in St. Louis. It's my first time here. It's definitely been really great exploring North Carolina and the campus you have, and exploring definitely local cuisines. I actually had some fried tomato the other day, which was to me a first time in my life. Never tried fried tomato before. I actually didn't know people fried them, right? But it was actually truly, truly delicious. So if there's any recommendations you guys have for me after the seminar for fried tomatoes, please come and see me. But I do have to say that the most spectacular tomato I ever had in my life is the one that you see here on this picture. Fried tomato, definitely close second. But this is actually a genome edited tomato. It's the high GABA tomato that many of you will probably have heard about. And it is commercially available in Japan. So it is edited in a way that it increases the GABA levels in the tomato, which leads to a lower blood pressure. And it is actually one example of the many, many really cool products that are being developed with genome editing technology. So there's GABA tomato, increased vitamin D tomato. We'll hear about Pairwise's mustard greens later today. And these are all being developed, in some cases are actually even commercial on local markets. And it's really, really great to see what this genome editing technology can do, and the potential that it has in bringing some of these really great products that are really aimed for consumers, as well as sustainability goals, being able to bring that forward in local markets. Now, one of the things you may have noticed is that None of these products are actually Bayer's products. We are working on developing our genome editing pipeline, and we're doing a lot of that through strategic investments with companies, investing in companies that are developing products that we think are key for sustainability goals in agriculture, like our partnerships with Pairwise, with CoverCress as well. We're investing a lot in collaborations, establishing partnerships where we can bring our regulatory, our R&D know-how into those collaborations. And we're continuously exploring more of those collaborations, more of those investments through some of our open innovation platforms that we have. That you might also be familiar with, like LEAPS, for example. We have our live hubs all over the world, for example, in Lyon, Grants for Ag, Test for Ag, so constantly looking for new partnerships. In addition to that, we also have quite a few licensing contracts, technology access that we leverage to have access to the genome editing technology that also support our in-house genome editing pipeline. Now, one of the things we have to consider, of course, as those products are being developed, as they're coming into the market, is what the regulatory landscape looks like. And this is part of what Natasha and I do on a day-to-day basis, is really monitor what does that policy landscape looks like. So for me, this is personally very exciting, but I do have to give you a heads up that I'm going to go into some hardcore regulatory stuff here. So there's exit routes in case anyone still wants to escape. But it is truly something that's important to keep an eye on. It's what we try to do at Bayer as well. You see this global map. It's been changing quite a bit over the last couple of years, because on the genome editing side, there's lots of developments constantly ongoing. I'll get back to this map later on in the slide deck as well. But I do want to focus a little bit on just 2 countries and walk through some of those examples as a test case as well. So just to give you some visibility on the complexity of the regulatory considerations. So I'll focus on Canada and the US. You see that they're marked here in green on this map. And what that means is that these are 2 countries where there's agencies that have in place specific exemption criteria that allow them to exempt genome edited plants from their GMO regulations. So if your plant fulfills those criteria that are being set in place by Canada and in the US, that means there's an opportunity to have it considered as conventional and not go through the GM pathway, which typically means very lengthy processes, heavy data requirements, and quite costly as well. Now, one of the elements as you dig deeper into the details here is that piece of complexity, as I mentioned. So the criteria for those exemptions are not the same between Canada and the US, and are not even the same in one country, which makes it a very, very difficult and very diverse landscape to navigate through. So the scope of exemptions is different. It becomes even more complex as you start comparing the different data requirements for those agencies, and even more complex if you add on the additional layer of predictability, which is also different. Some of these frameworks are a little bit more mature than others. So that means predictability is higher typically. So let's focus on the US first. And I'm going to talk mostly about just the scope of the exemptions, that scope component, just because of lack of time, I think, but definitely feel free to look at the slides afterwards, and happy to catch up on more details on data requirements and predictability as well. So if you look at the US, there's actually 3 agencies that can have oversight of genome edited plants. I'm not going to talk about FDA just because they don't have a separate policy for edited plants in place right now. But I know Dan is going to talk about FDA later on in his presentation a little bit as well. So if you look at USDA, they have oversight of all genetically engineered plants. So that means genome edited plants as well. And they do that, Natasha already mentioned Part 340. So that's their regulation for edits and the exemptions that they have put in place. And that came out in 2020, so a couple of years ago. And then you have EPA, and they regulate pesticides or pesticidal substances. Which also includes pesticides produced by the plants, which we refer to as plant incorporated protectants, or short PIPs. And they have exemptions in place for genome edited PIPs as well. They came out with their final rule in 2023, so back in July this year. Now, what are those exemptions that they have put in place? And this is where it becomes very regulatory-y. But essentially, if you look at the USDA policy, one key threshold to keep in mind is that they only allow one edit to be exempt. So if you have an edited plant with 2 edits, by default, USDA will consider it consider it regulated, and you're not exempt from their regulations. If you do have a plant with 1 edit, that edit has to fulfill any of those 4 criteria in the table. You can have a repair of a non-templated targeted DNA break, so SDN1 type of edit. You can also have a single nucleotide change. You can have a cisgenic insert or modification. And the last one is not related to the type of edit, but is really more related to familiarity. So if the USDA is familiar with your plant trait mode of action, they won't review it again. And so you're also considered exempt from their regulations. So this one applies for genome edits, but can also apply for GM. Now, if you look at the USDA, there's no threshold. You can have multiple edits in one plant. But those edits have to fulfill either one of those 2 criteria from the EPA. And the EPA's criteria is really focused on edits, essentially knockouts, that decrease or eliminate activity of your gene, which in and of itself leads to a pesticidal effect. And then they also have another exemption criteria, which is similar to the one at USDA, that allows for modification, insertion of sequences that then lead to a sequence that's already known to be present in the plant's gene pool. So I know I covered a lot of different elements here, but that's just to show the complexity of the regulatory landscape in the US. You have 2 agencies that could potentially regulate your plant, but they have different exemption criteria. So that could lead to a situation where you have a plant that is considered regulated by one of the agencies, but considered exempt by the other agency. So it's really key to know what those criteria are. As I mentioned, I'm not going to go through all the data requirements, just know that they're all available as well. It's typically less data heavy compared to the GM data requirements. And there's also some differences in terms of predictability. USDA has their policy in place since 2020. They've been doing evaluations for a lot of years. So there's quite a few exemptions that already gone through or quite a few products already gone through the process compared to EPA, which is, to my knowledge, I don't think there's been any product that's gone through the EPA exemption route. If you look at Canada, which was the second country I wanted to highlight here as an example, it's a similar situation. There's multiple agencies that could have oversight of genome edited plants. I'm not going to talk about the feed agency here because they're still developing their policy. It's actually under public consultation, I think, still. So I'll talk about Health Canada and CFIA for environmental release. And so both have policies in place. Health Canada have had their policy in place since 22, and then CFIA since this year. And Health Canada really looks at food safety, whereas CFIA looks at environmental safety for environmental release. And so looking at their scopes of exemptions, this definitely does feel like a little bit of a breath of fresh air because it is pretty much aligned. You don't have different boxes like you have in the US. They take a very similar approach. There's no thresholds for number of modifications, number of edits. And their key criteria is presence or absence of foreign DNA. So instead of looking at the type of edit, they really focus more on the end product. What is your plant characteristic? So presence or absence of foreign DNA is key. And then Health Canada, because of their scope of food safety, will also look at any potentially altered food safety characteristics. And then CFIA on their environmental side looks at environmentally potentially impactful characteristics as well. So this is an example, I think, of a country where, again, there's multiple agencies, but they're pretty well aligned, which makes it easier for developers to navigate through that regulatory system. Again, some differences in data requirements, and then some difference as well in level of predictability, where Health Canada is definitely a little bit more experienced compared to CFIA, considering their policy was adopted last year. So trying to put all the dots together, going through a case study. So let's say if you have a soybean plant, you want to commercialize a soybean plant here in the United States, and you want to do that. You want the trait is virus resistance, where you want to focus on. You want to make it resistant to a specific virus, and you're trying to pursue to do that with two approaches. One on the left-hand side is editing. The other on the right-hand side is a transgenic approach. On the editing side, you work with simple edits. You do a knockout in a virus receptor. The gene no longer expresses or is no longer active. No protein is expressed and no binding happens, which leads to your virus resistance. And you do that by SDN1, let's say. You don't use a template. On the right-hand side, you have your transgenic approach. You insert a gene from a non-sexually compatible species, let's say a bacteria, that then leads to your virus resistance. So let's say two approaches. What does that mean? Now, I mentioned I want to cultivate in the US, right? So first agency I would think about is USDA because they have oversight of all genome-edited plants. So what about USDA? Well, I comply with the threshold. I only have one edit, and it is a non-templated edit, right? So I didn't use a template. Great. I would assume I'm considered exempt by the USDA and can go through that confirmation process. What about EPA then? Am I considered a pesticide by EPA? Well, your trait is virus resistance. Viruses are pests. So EPA is definitely something to consider for this type of product, and you'll probably be regulated under EPA. What does that mean for my EPA exemptions then? Well, no threshold, great. So I checked that box. And it is in fact an edit that leads to elimination of gene activity, which then leads to direct pesticidal effects. So I also checked that box at EPA, which means I'm also very likely considered to be exempt and can go through that confirmation process. Now, the other thing to consider is what about Health Canada, or what about Canada? Neighboring countries could be that I'm using my product ends up in the food supply. Should I consider Canadian regulatory system? Probably you want to have a look at Health Canada, right? In this case, no threshold, no limits on number of modifications, great. I have no foreign DNA because I deleted a part of a gene and I have a knockout. And the plant does not have any altered food safety characteristics because it is a virus-resistant trait. So all green marks here on the regulatory front for this particular edit, but what about my transgenic insertion? I can go through this rather fast because you see that in this case, let's say I don't have a familiar plant trait mode of action at USDA, I won't be considered exempt from the regulation and go through the RSR process. At EPA, it's not an edit, it's a transgene insertion. So by default, you don't qualify for the exemptions. And then Health Canada, you do have foreign DNA. You insert a bacterial gene, so you don't qualify for exemptions. And you go through the assessment at Health Canada as well. That definitely makes a key difference in terms of costs, as we had mentioned earlier, but also makes a key difference in terms of timelines. If you use that editing approach, it can really make your timeline much shorter, which is definitely an advantage and can make a difference in terms of determining what approach you want to use. If you look at the GM side, and I think these timelines are still quite optimistic, but you can ensure, or you can really expect that process will take a couple of years. And this is just for the US and for Canada in this case. One thing to consider is you're working with soybean here. So where does it go globally, right? It's highly traded commodities. So what do I do with all these export countries and what's their regulatory system look like? So that's where I really have to think about what are the key export markets. In the case of soybean, you can look it up. There's different sources where you can find some of those materials and those numbers. Looking at the USDA source, China, Mexico, EU. Well, that brings us back to our map, right? EU, everything considered GMO. So you have to go through your GMO pathway. Mexico, not defined yet. It's under discussion. That's why it's blue on our map. So the pathway will likely also be the GM path. And then in China, it's a very GMO-like process, which means in-country field requirements still, in-country field studies, a lot of food and feed safety requirements, and you're looking at a couple of years probably as well in China. So that's another consideration depending on really the crop that you're working with. So this was definitely not to not to scare you all, but I think it's really something to keep in mind as you think about some of these regulatory complexities and just the importance of knowing what the regulations are, what the exemptions are. It's a complex puzzle, but it's not impossible to find your way. It's take some time, have to dig into the details as well, and really think about even the first question, is my plant even in scope of the regulation? Should I even go through this agency? That's not always a given, right? That's not always certain. And if you are, then really think about looking at those data requirements. What's the process look like? And really also stick to the data requirements. Because if you provide too much or not enough, it can lead to lengthier timelines, of course, which are something you would want to avoid using an editing approach as well. One thing to keep in mind for those timelines too is that they're sometimes prescribed in the regulations, sometimes they're not, but also realize that the theoretical timelines do not always necessarily align with the practical ones. So that's an important consideration as well. But I do think there's a key opportunity here with editing technology, and especially if you look at all the products that are being developed, like we talked about earlier, there's great products coming into the market. And if you look at the numbers on USDA website and also in publications in Argentina, we see that really small developers are putting their products through those systems. It's local companies, it's public researchers, it's not the big multinationals like we've always seen with the GM and the biotech space. And I think that's a key opportunity here that we have, and that we need to make sure we don't lose and continue advocating for regulatory harmonization, which will definitely make it easier. And to end, just to leave you with a couple of tons of resources. I have plenty more, so let me know if you need any more information. But I think these are a couple of key ones that I can think of for you to have a look at. If you have any questions and want to dive even more into the details, you'll have these as well at the end of the presentation. With that, I want to really thank you for your attention. I hope I didn't bore you too much with too many regulatory stuff, but really thanks for the opportunity to be here, and I look forward to the Q&A session later.
Thanks, Lieselot. And our next speaker is from one of those slightly smaller companies than Bayer. Dan Jenkins is going to join us from one of our local companies, which is really pioneering this technology in plants. So please come and join us, Dan, and thanks for being here.
One moment with your slides.
Yep. I'm just watching all kinds of magic here. Okay. Why don't we just go to the next slide? Thanks. All right. Hi. My name is Dan, and I lead regulatory and government affairs and quality for Pairwise. And we're still working on getting our slides up there. So Pairwise is a company that was founded about 6 or 7 years ago, and we were founded by some of the inventors of CRISPR and the technology from MIT and from Harvard and Massachusetts General Hospital. Pairwise is a health-focused company. So our mission, the idea here is, how do we get people to eat more fruits and vegetables, right? And we think that the way to do that is to do things like make them seedless, right? So we are a company that's not only working in produce as you traditionally think of it, and some of these crops, we're also working in trees. So cherry trees, for example, making a pitless cherry. We got about 160 people, most of which are here, site in RTP, site in Durham, and site out in California. And right now I'm afraid to touch this and try to advance these slides because that was a lot of work to get up there. I'm going to try. Oh, it did work. Great. Okay. So again, we're working on developing these new varieties of fruits and vegetables using gene editing technology. And as I already mentioned, this is our mission. So we're trying to use CRISPR to break down these barriers that prevent us from eating as many fruits and vegetables as we should. Now, barriers can mean a lot of things here, But what we're talking about is just making them a more attractive choice when they're there in the grocery store, right? So the idea is to get people to maybe turn a little bit more often to the right to go to produce versus to the left to go to the chips. Okay, because our mission is to address the problem that we have not only in this country, it is global, in obesity and all the health issues related to that and non-communicable disease related to diet. And the way to do this is to hopefully present something that is attractive to improve people's diets. Almost nobody in the country eats the recommended daily amount of fruits and vegetables, except for really vegetarians. So in terms of commercialization in this pathway, there's a lot of things to think about beyond just the regulatory. But the regulatory, if it's not there, you're not going to get started. I mean, obviously, you want to be legal, right? So when we're looking at different areas around the world for commercialization, there's a lot of things to take into account. And it's not just the biotech regulatory, right? So as we begin to explore places to grow these things for counter season, or because the regulatory systems are very difficult, you also have to have the regulatory in place for shipping the plants down there and bringing the fruit back. That's a lot of regulation in and of itself. And then when you get into a country, you get past the biotech regulations, you start getting into the regulations of produce, fruits and vegetables, labeling, food safety. There's a lot. So we go past that farm gate, and there's all that other body of regulation to think through. And of course, these other things, it's got to grow well there. Want intellectual property protection? Are they members of UPOV as a country? One of the most important things is, can we find a grower that is really high quality, that really cares and pays attention, so that we can have the commercial success that we need? Of course, the cost of goods is important, and just the infrastructure. How quickly and how well can they get that produce cold and maintain it and ship it while meeting import requirements from FDA and USDA? So there's a lot to consider here. So I would say that the global outlook for gene editing is really positive when it comes to regulatory, especially when, like Lieselot just did, we compare it to GMO, which is a place where I came from, and we probably all have a lot of scars from that. And I love what the impetus for this is, which is that many countries are looking at this because they recognize that there are problems that we have to solve. And I would say, in terms of the opening talk and acceptance, that what we see in the data is that the younger you go, the more accepting they are. I think there's a lot of concern about a lot of problems that we have, and they look to these technologies as being something that can really help. The issues with acceptance tend to be with my generation and older. And I think that we see a positive outlook there as well. So, and, you know, in particular, as was just being mentioned at the end of the last talk, this opportunity for smaller and medium-sized companies such as ours, and working in specialty crops is the aspirational goal here, and also cited as part of the impetus for making these policy changes. Another map, okay? So things are generally green on this map. But if you dig a little bit underneath it, it's not all 72 and sunny. There are some issues, as was just being pointed out. So what I want to do here is say the general trend is absolutely positive. That's the good news. And the way countries are looking at this, they can come from different angles. So in some places, very focused on what is the process that you use to make the product, while at the other end of the seesaw is, well, we're focused on product. So Canada, for example, is at the side on the product base. What matters is the characteristics of the product and the environment you're introducing it into in terms of a risk assessment. It has nothing to do with how you made it. I'd say the US and some of the things going on here are actually more to the other side. You use gene editing. This is a European approach as well. This is where you see the UK has kind of made a turn recently and getting away from more process-based. And then once they make this sort of fundamental opening philosophical decision about how they're going to start, how much of a haircut do they give it, right? So is it going to be like conventional, or like conventional plus a little more? Or is it going to be like GMO and like GMO light? And this is why we see differences in countries around the world. So this is updated from a paper that we published in Nature Plants this past spring, where we were looking at really what is the utility almost of these actual systems? How do they really treat things? So when we're in USDA, and if I could make that little red dot in the upper left a little smaller, I would, but then you wouldn't see it. So Lieselot mentioned this is the exemption here is for a single edit, but it's actually for a single edit in a diploid. So you're going to be fine with disease-resistant soybean. But if we were to do disease resistance in potatoes, which are a tetraploid, strawberries, which are an octoploid, you don't get this exemption right now with USDA. So you're in that big red circle over to the right, which means you're basically put in the same bucket as GMO. It's not necessarily a terrible bucket. It's just you're in the same bucket. And it's not quite so predictable right now on that scale because it's so new. We don't know how it'll really go. There's different steps to the process and what happens if you have to go to the second step. What you see in the upper left in terms of being very predictable and basically treating you like conventional are places like Canada and most of Latin America. So when I look at some of the statistics on some of these things, like all those confirmations of exemption at USDA, my company is a quarter of those. And when you look at the determinations that have occurred in Latin America, my company is a fair amount of those as well. And of the 4 that have gone through Canada, well, we're one of those. We're 25%, we're pretty consistent. But what I see here is there are differences that have to be worked through. And so when you're thinking globally as a company and launching products, where to cultivate, where to sell, you have to think about this because it means expense. So, and why, right? So this is from our paper as well. Like we can create a seedless grape. My kids don't know that seeds are in grapes because they've never had them, right? We could recapitulate this mutation using multiple approaches, using gene editing, whether that's templated repair or just your simple knockouts, and they would be treated differently than the exact same thing and differently compared to each other, depending on where they are in the world. So then that's driving your choices about what products you're going to launch and how you're going to approach them. It's already narrowing things down. Okay? And one of the challenges that you have in working in these kind of specialty crops is that building up transformation systems and doing all the testing you need to do to make sure it's working goes well past the farm gate. We actually have to have consumer testing as well, which goes on for another year or two. And then how does it work in a bag and respire and get shelf life? So there's a big challenge there. And all of this adds up to, do I put $1 into it or not? So we're launching this product. This is called Conscious Greens. This is an edited mustard green. Mustard greens taste bad. Okay, so we made them not taste bad. And some folks here tried them yesterday and they can attest to this. So they've got a huge wasabi flavor, which is actually, for some people, they really like that spiciness, but you don't want to eat a bowl of it. And they're really nutrient dense, but unpalatable. And there's great aspects of these brassicas, which is what they are. Besides being nutrient dense, there are ones that look exactly like spinach, exactly like romaine. They are purple, they are red, they are frilly, they are broadleafed. They're naturally resistant to viruses that sent lettuce up by like 10x in terms of a cost per crate basis because it ravaged California a year or two ago. So this is what we're doing. This is, and it's in terms of its gene pool and what you could do with breeding. The gene pool is massive. All these brassicas interbreed. You've got your diploids on the corners and your tetraploids when they come together in between them. And so this gene pool and what's available is pretty substantial. So most things that you're doing here, you could probably do through conventional breeding. It would just be hard and take a long time, which is the point of CRISPR, right? One of the things to think about here is just the advent of the technology means that it suddenly becomes worthwhile to invest in these crops and these traits and specialty crops, which are orphaned in terms of investment, because you could actually do it and get there quickly. Before you couldn't. So, and you can buy all of these things. Many of them actually have this mutation in them that makes them not taste bad, not to have this wasabi flavor. So these have a history of safe consumption. And we published all this. So if you want to go look it up and see what we did, it was This is a good example of working in specialty crops. We thought maybe there was about 5 edits that we had to do. Turns out there were 17 because these genomes are not well known and they're not well annotated. So, so then we started to go through the regulatory systems. And we got this through under the old rule at USDA. And under the old rule, which is the top of this slide, about 135 products went through that. That's a little deceiving. So what really happened is that once industry and academics too knew that the new rule was coming, remember that big red circle on how most of the stuff would be treated. I would say that probably 70 products went through the old system in the first 28 years, and then the next 60 to 70 went through in the last 2 years. People pushed them to get them out. I will tell you that we did ours in 2 weeks just to get it under the old rule. It came back out in 60 days. Now we have a product to sell. Under the new rule, it takes a long time. It takes a long time because USDA, who wants to do the right thing and absolutely wants to see this innovation succeed, they have written a rule where everything gets captured in the room and there's one tiny window out, single edit in a diploid. So now they've got a lot of work and it just keeps getting bigger because this technology is being used by so many people to do so many things in so many different crops that they can't handle it. So USDA knows that and they're working on it. That's the good news, but that's where we are. And so under the new process, I will just summarize to say it takes a long time. It's taking a long time. It's taking as long as it took for GMO. The data requirements are much less. That's great. But this is where we are. One thing I would point out, though, that's particularly critical for a smaller company like us is that time that you're waiting means time that you have to be under permit. And just finding a cooperator to say, I'll grow your mustard greens in Yuma, Arizona, knowing that the US Department of Agriculture can come and inspect me, because this is biotechnology, is very shocking. And they then adjust the price by 5 to 12x. And so what I was explaining to some other folks yesterday was, it's probably $800 to grow an acre of corn. It is $80,000 to grow an acre of blackberries in the central coast of California. So all this time under permit means expenses going through the roof, and it's very difficult to do this work. So we got to get out of here, which makes me start looking towards what can we do in other countries. So you'll see that USDA has gone, therefore, from having maybe, my guess is, I don't know, 60 or 70 permits for plants a year in the past for GMOs. They have over 300 now. So, but again, they're working on it. Now, FDA, you're not required to go through FDA. They do have a GMO process there, which they call BNF, Biotech Notification process. We went to them and just said, we're going to do an informational seminar. We're going to disclose a lot of the things that I just showed you. We have a publication that shared what we did. We're going to talk about the history of safe use. FDA still had a lot of questions. They responded to us with a letter saying that we did this. That way, FDA at least was informed, because this is something we could have done through conventional breeding. So we didn't feel that we should go through a GMO process. My philosophy, our philosophy at Pairwise, being a leading edge here, is we're very much interested in setting precedents that are the right ones in terms of data requirements for something that could have been achieved through conventional breeding. Health Canada was mentioned. This is my favorite story. We went to Health Canada, and Health Canada had already had another company go through. And sometimes we're our own worst enemy. And we have a tendency to say, yes, we'll give you all that bioinformatic analysis. Yes, we'll do that compositional analysis. Yes, we're gonna give you all that allergen tox analysis. Very much GMO, which sets a precedent. So we came in and got asked for all those questions, and no, we're not going to do all that for something that could have been done through conventional breeding. And in fantastic Canadian style, they said, sorry. And we had a wonderful conversation with them. And so then we worked our way through it, and we pressed submit at 8 in the morning, and we had an answer back that we were novel by 6 that night, so 10 hours. And love it, right? So again, super positive. Similar in Latin America. So we have moved this product through determinations. Guatemala took 2 weeks. Colombia, so Honduras was really quick. Colombia was a little bit longer, but that was because of us. We're just about done in Chile. That'll probably take about 4 weeks. So when you start going outside the US where the requirements are non-transgenic, tell us about your product, much faster, much more efficient. We've already started investing in these countries because of it. So this has been really positive, and I'll stop there. So thanks.
So our last speaker is from the university, so from the Office of Research Commercialization, Koltarin Schoen. And Koltarin really runs all of the IP systems here at the university. Before I hand over to him, I think we're going to go a little bit over. I don't know if the panel can stick around because I would really love just to have a few minutes for any questions you have. But after Koltarin, if anyone needs to sneak out, then I'll give you an opportunity because I know it's everyone has other stuff that's going on. But first of all, Koltarin, thanks a lot for being here.
Thanks, Adrian. Hello, everyone. The title of my talk today is going to be Commercialization of Early Stage Research. So this really is looking at taking a step back before you get into the path for commercialization, or you're thinking about what regulatory pathway you have to consider. So what can we do before that to ensure that if you're interested in seeing your discovery get to the marketplace, what can we do to enable that or make it more likely to be successful? So the way I've set this up is I've listed a few questions that I think would be relevant to the audience, and I'll go through those as I go through the talk. So at a high level, what is research commercialization or tech transfer within a university setting? What does that mean? What are IP assets? For example, patents. Why are they relevant? Should we care about them? Are they important in the research commercialization process? And then finally, how do universities engage in research commercialization? So, as a university, all of us are very familiar with what universities do. We train, we educate, we teach, we do research, we disseminate a lot of that information. But as I'm sure a lot of you are seeing, universities are starting to play a more bigger role in innovation and entrepreneurship, right? Innovation around research discoveries and trying to see those discoveries move out from the lab to the marketplace. And then entrepreneurship, like something what Jack is doing and what he talked about, where faculty want to get more involved beyond just that early discovery phase. So a lot of what I'm going to talk about are general. There are always exceptions to what I say, but generally as a university, we don't make products, right? So what I mean by that is we have research discoveries occurring at the university, but we don't go through the whole path from discovery to launch. It really is that early stage. Around research discovery. So we don't make products. Then how do you bring them to the marketplace? That generally occurs through licensing. So what are we going to license? So you're licensing the research discovery. How do you license the research discovery? That is basically developing an IP asset around it or protecting that research discovery. That is then going to be licensed to whom? Generally, it's either an existing company, and by existing company, it could be a big company like Bayer, or it could be a relatively smaller company like Pairwise. And there are going to be different considerations that go into that. Or it could be through the formation of a startup company. And this is where a company is formed primarily with the goal to license IP out of the university and then further de-risk it, where maybe a bigger company now would be interested in getting access to it. So that would be an example of what Jack and his team has done. So IP assets, again, in the interest of time, I'm not going to go into details around this. I'm sure we'll have seminars later on or workshops where we can talk about different types of IP assets, why they're valuable. But generally, there are different categories of patents, copyrights, tangible material, know-how, and trademarks. I'm going to talk a little bit about patents because generally that is the path universities take to protect innovations that are occurring at a university. In order to obtain a patent, it has to be patentable subject matter. It has to be useful. Your invention has to be novel, non-obvious, and adequate description of that. Again, we can get into discussions around what that means, but that is a lot of assessment that our office will do when we work with faculty to ensure we meet those criteria, and then we can go in and obtain a patent. So then the question becomes, why seek IP protection? Why should we go around doing that? Is it important? So from a university perspective, when we look at IP, and again, I'm talking about patents, we really look at them as a critical tool to enable commercialization. They're a tool in that process. What it does enable our faculty is, as we all know, publishing, talking about your research is critical to what you do. I say faculty, graduate students, postdocs, to everyone. So how do you protect that innovation and still talk about it without losing rights in that? And that's where patents can play a very important role. So long as we file a patent before you talk about your innovation, you can still protect it. And that still can then be used as a tool to enable transferring that discovery from the lab, hopefully to the marketplace. So, at a high level, I mentioned stuff that happens at a university, or the research that's happening is really early stage. What does that mean? That means it's probably going to take, depending on the technology, anywhere from 3 years to 10 years before that's going to be converted into a product and brought to the market. And again, depending on the technology, it can take millions of dollars of further research and development beyond the discovery phase to convert that into a product or a service. So how do you incentivize someone to take that risk? How do you incentivize someone to come in and put that money into developing that product, knowing that if they are successful, someone else can come and copy that or make a generic version at a much lower cost, which would allow them to sell it at a lower cost and can then prevent the risk taker from recouping their cost and making a profit on that product, right? And that's where IP, and again, patents can be very critical and very important, because they do ensure, hopefully, they will ensure that if you have taken a risk on developing a technology into a product, invested millions of dollars, you can then recoup your cost and make a profit during the term of that patent. So that's why patents can be very important. Just to give you a quick example, one big success at NC State was a small molecule called 1-MCP. It was a molecule that binds ethylene receptors, prevents ripening. This was a technology that was developed in the late 90s, got licensed into a startup company, because again, it was very early stage. It took about 5 years to go through a regulatory process and get approved. And then it was sold in the market, and it brought in close to $30 million in royalties back to the university. And this is not talking about the benefit it gave to the company that was able to sell that product. Now, if that was not patented, if those compounds were not patented early on, it is very unlikely that any company would have invested the time and effort required to develop that into a product and take it out of the lab. So it would have been a great research article. It would have been great a publication, but it never would have been, most likely would not have been converted into a product that came to the market. So just high level things that we do within our office. So our Office of Research Commercialization, we manage all IP at the university. That's what we do. All of us as employees are required to assign our IP to the university. And then the university looks at that, and we try to be the best stewards we can be of that in terms of managing it and then making it available to industry partners and others, hopefully resulting in products to the market. So when we get a disclosure, what we're doing a quick assessment to see if it is worth filing a patent. So I talked about patents. It generally will take about 3 to 5 years to get a patent issued. It will cost about $20,000 to $30,000 per jurisdiction to get a patent issued. So if you're filing in the US, you talk about 20, 30,000. You go to Europe, you keep on adding that up. So it can get very expensive. One of the requirements to get a, when we look at the requirements to get a patent, it says patentable subject matter, novel, non-obvious, useful, but it doesn't say, the patent office doesn't care if there's any commercial opportunity or if it's worth anything. They do not care. right? So you could get a piece of paper that's worth $20,000, but has no commercial opportunity tied to it, right? So what we are trying to assess from our office is to see if there is a need, right? Is there a market need enough to justify spending $20,000 on that technology? So we're not making a judgment on your research at all. It really is to see if there's a market need and a path to move forward. So that goes into, what is the problem we're trying to solve? Who cares about that problem? What is the stage of development? Because that will determine who you potentially can license it to, or even if you can license it. What is the path of the market, the technology to market, right? And a lot of things talked today about was the regulatory path, and that can be a key factor. It's good to know upfront what is your path, or at least have a good idea of how you're going to bring that to market, or a partner can bring it to market. Because if there isn't a clear path, it's very unlikely that we'll be able to find a partner who's going to be willing to take it forward. Other factors are things that we look at in terms of determining licensing strategy, but market consideration, right? Who cares about this problem? Is it a good to have, or is it a must have? How big is the market size, right? So if it's going to take you $20 million to go through regulatory approval, and the market size is $1 million, you're not going to find a partner who's going to move it forward and take that risk. So what is the market opportunity for the technology, and is it worth protecting? Again, this is all business. Again, we're doing a very high level early stage assessment. We're not doing it at the same level that companies would do it, because they have, by the time I think companies have these and are going through the regulatory, they know what the final product is going to look like. They have the data. They have a lot of data and information that allows them to do appropriate finances and figure out what it's going to cost. From a university perspective, we're really doing a high level early assessment, which we know might not stand out to be true at the end of the day, but we still have to do an assessment to see, is there an opportunity big enough to justify spending $20,000 on a patent? All of these are questions that we look at. Then IP consideration. I know microbes were talked about. Is germplasm a key question? Is who owns that? But if a faculty member at NC State goes and gets a microbe, where did they get it from? Does the university actually own it? Can we license it? Because you could do great research on it. It could be the best microbe in terms of the application. But if you got it from a place that is not NC State or owned by NC State, we can't license it, or we can't make it available. So those are the type of things that our office will assist in assessing and give recommendations on how to deal with those issues. That's around tangible property. Similarly, around patents. How broad are your claims? Is it easy to design around your patent? If a potential partner is going to license a patent and pay a cost for that, what value does it give them? Is it easy to prove infringement if someone is infringing your product? Or is it going to be very difficult? So those are the type of considerations that our office will go through in assisting our faculty, and then as we do deals with startup companies or more established industry partners. Okay. The licensing considerations, again, very similar. Is it jointly owned? Who owns the IP? These are due diligence steps that we do. Hopefully, these are steps that we do, and this gives comfort to industry as they come and try to license the technology, because they would want to make sure that the university owns what they're licensing. The statements that they're making are true. The representations they're making are true. It gives confidence, because there's going to be a lot of investment that goes in developing those technologies into products. And what you don't want to do is, once you're ready for launch, you find out that you didn't have certain rights that you thought you did in those technologies. Okay. So why should researchers at NC State care about it? It can be very rewarding in terms of social impact, professional development, but also monetarily. So at NC State and other research universities, we are required under federal law to have a patent policy under which revenues that come back, a portion of that need to go back to the inventors. And this is primarily to incentivize our researchers and inventors to work with our office, spend the time that it takes, hopefully, that will result maybe 3, 5, 6 years down the line in some monetary return for all that time and effort. So as I talked about that one big success, if the university got 30 million, just imagine a big portion of that went back to the inventors as personal income. It raises the visibility of the university in terms of what type of research is occurring, the expertise that the university has. It creates funding opportunities for our faculty members, opportunities to collaborate with industry. So I'm very proud we have collaborations with all the industry partners that have talked today at NC State. So and IP is going to be a key aspect in those collaborations. It helps us retain and recruit faculty. So, Jack, for example, is doing great work. Do we want him to ever leave NC State? No. What is going to incentivize him to stay here more is the resources we offer him and the opportunities we give him to do more than just the research, right? Engage with the startup company he's working on. And at the end of the day, from a university perspective, it really is to stimulate economic growth in the region, provide job opportunities, and to benefit the public. So key aspects, and prior to licensing, again, this is all stuff we do. What are we licensing? What is the value? Value proposition is key. If we're going to license it or talk to industry, why would they care about our technology or research discovery? Why does it matter? Stage of development, how is the technology protected? Due diligence on IP, then finding an appropriate licensee. So at the end of the day, again, it's a lot of information, but that's why we are here. We are here to assist our faculty in going through that process. So what I would encourage everyone, as you're coming up with research discoveries, please reach out to our office. Don't think about whether it's too early. Anytime, just reach out. It's better to talk with us early than later once you're ready to publish or present. And with that, I have questions.
Thanks. So guys, I realize as I predicted that we've gone a little bit over. I'm looking at the potential panelists, desperately hoping you could stay for 15 more minutes, and hopefully those of you in the audience can stay, as many or all of you, and we're going to have a good conversation now, and hopefully there's some questions that's going to come either from the NC State or from many of our industry partners in the room. So guys, would you like to come up? And while you're doing that, I want to say a big thank you to Kathleen, who put this all together. She was meant to be the MC. This would have run on time if it was her doing it, but she's lost her voice. And so I stood in, and consequently, we've gone over 15 minutes. But so thank you, Kathleen, for putting it together. And also there's some probably lukewarm coffee and some, unfortunately, sorry, Dan, some very unhealthy snacks over there, but you're welcome to go for them if you'd like. And we're going to be very informal. So I'm going to open it up for questions or for comments for any or all of the panelists. I have one myself if no one else wants to go, but please, Danisha, why don't you go ahead?
Somebody has to start, right? Hi, Danisha Seth Carley. Y'all know me. I'm the director for SERS. I'm also faculty in the horticulture department. And I have a question, Dan, for you specifically. I'm going to wander around, but I'll try and rein it in relatively quickly. Can you all hear me? I don't need to use a microphone. Thank you. So you said something that really sparked my excitement and my interest, and you said that not only do especially Americans not eat enough fruits and vegetables, and that y'all want to help correct that, but also make the gap between having fruits and vegetables accessible to communities smaller, right? Sounds to me like what you're doing costs a lot of money. My guess is that will then get translated back to the consumer. How do people who are struggling to just, pay their rent afford to buy fresh fruits and vegetables from, say, Food Lion rather than Whole Foods? Because as a single mom, I struggle with that myself, even though I get paid relatively well by NC State. So how do you approach making it not only more accessible, but also cost effective or cost accessible for communities? Thank you.
Do I need to use a mic, or can you?
I can hear you.
Okay, I'm trying to speak loud.
I'm the only important one, right?
Okay, Natasha, I'm right here.
Can you guys hear over there? Sorry.
As long as we've got each other, right? That's all right. I don't honestly, when it comes to breaking down those barriers, what you're talking there is about the socioeconomic ones, and I don't know if we're going to be able to solve that. In terms of how we're trying to approach pricing on these things, look, a lot of these will be more of a premium price, at least to start. And so, our immediate goals are to get these varieties in there, demonstrate acceptance, have initial commercial success, and then hopefully come in behind that with other types of traits that can be applied more broadly and perhaps can address that one as well by, if you talk to folks in this area of agriculture, their top 10 issues are labor. So if you could do something in terms of improving like plant architecture to mechanize harvesting, then maybe that helps with costs. And maybe we can get there, but that's not where we're starting. We're starting with something that we think would have direct appeal to consumers so that we can then come behind with that later once that acceptance has been demonstrated, and then maybe we can get there. But anyway.
Thank you. And I didn't expect you to solve everything all at once, but thank you.
We have another question, please.
As a follow-up, I heard, I'm Sherry Witt from BASF, by the way.
Hi, sure.
I heard that the conscious greens mustards will be available in the grocery stores.
Yes.
So how soon and where can we buy it?
So we've had a small commercial launch in food service. So it's been in like some university cafeterias, some restaurants, mostly up in the Northeast. In terms of the retail launch, fingers crossed, probably here in first quarter. It's probably going to start on the West Coast. They're grown on the West Coast. Shipping is easier, right? So hopefully soon.
Yeah, fingers crossed.
Yeah.
So another question. So I was curious if Bayer and Pairwise, if you support the, I want to say it's the Center for Food Integrities. It has a framework for the responsible use of genome edited products. So are you as companies getting behind that initiative or?
Yeah, I'm going to use a microphone because I'm soft. I speak soft, but yes, indeed, it's something that we're looking into as industry, the CFI network. So that is really a framework that will provide, or that is already launched, and that's providing a framework in terms of stewardship responsibilities and transparency commitment, et cetera, and companies can sign up to it and be part of that framework. So we haven't signed up as Bayer, but it's something we are looking into. There is actually going to be another framework that's currently being developed as well that's more globally focused called the GSG framework, which is similar to the ETS one that exists also for GM. And that's something we're also actively engaged in discussions. We have different colleagues involved in those conversations and shaping up those frameworks to have that launched, I believe, sometime soon. So we're definitely keeping an eye out on those and assessing what would be necessary to actually sign up for those and be part of those programs.
So I'd say similar, keeping our eye on it. I don't know if we'll sign up for it anytime soon. I'm gonna get a little bit wonky here. So I think a program like that is probably more important and more relevant for grain trade. And when it comes to produce and those supply chains, the feedback that we get, whether you're talking to an executive vice president who selects all the produce that we eat because he or she is the buyer for Walmart, or he or she is the buyer for name your grocery store chain like Kroger or whatever. One of their main points is don't do anything that changes what we already do. Don't disrupt our supply chain. Don't add on additional things. And that's a hassle and they don't need much of a hassle for them to turn away. And I also get concerned about something called the responsible use for gene editing, and the impression it gives that somehow that program is addressing a safety concern. It's not. I think for us, the principles of it, transparency, having a stewardship program, all the things that go along with basically a quality management system are the right things to do. And we're doing those. So for example, on the back of our packaging, get better flavor through CRISPR, which I think is cute. And then there's a QR code and like talk about the technology. So all that transparency is there. But we have concerns about, at least with produce and fruits and vegetables, what it could mean. But as long as we're doing those things, then yeah.
Jack.
My name is Jack Boyne, and NC State grad. And first of all, your presentations were really interesting. Thank you for being here today and allowing us to listen. My question really is, with all the new technologies that are driving toward more sustainable agriculture, yet we still seem to have the same timelines for many things that were the classic pesticide development that we saw 60 years ago or more. So I guess this gets to Jack's point early on about acceptance, and why aren't we moving the needle on that? Why don't we, with every new technology, it seems to me that we're still stuck in the Rachel Carson time zone of this is technology is bad. And how do we affect that either as a university or as a company employees?
Can you hear me? Okay.
Yep.
So I have my thoughts and theories on that. One of the things we've had conversations as my colleague has come into town over the last couple of days is how we talk about the technologies, right? So we talk about learnings from the GM, the way it was talked about with genetically modified plants, right? And how that scared a lot of people when you said genetically modified. So hopefully, as we're doing new technologies, especially with the platform that I work with Microbes, that we can talk to the consumers, to the people who need to know the answers on their level. And we talk about science education, like at Bayer, we know our science is good, same with Pairwise and other companies in the room. Communicating science, especially globally in the United States, as we all saw in the last couple of years, is very hard, right? Your average, I think we said yesterday, the average science education is 6th grade. You're talking, and then we're talking CRISPR, we're talking edited microbes, and it just goes over their heads. So we as an industry, as our academic partners, we really have to figure out how to properly talk about the technology and put it in ways that your average consumer can understand what's going on and being transparent, but also really communicating the science on a basic level. I think that's where we start to get into societal acceptance. I think it's really cute about, you know, made by CRISPR on the back of because again, transparency is key. And I think when you get into that I think part of the problem with GM plants at the time was the transparency wasn't as clear maybe as it is now on how we steward these products. So that's just how I think we can do some societal acceptance.
And I think maybe just one thing to add to is that at the end of the day, you know, Natasha and I are part of Bayer, right? So big multinational company, and we do a lot of outreach activities. We publish our data, but at the end of the day, we still feel like there's not a lot of trust in Bayer per se. And we see that when we have regulators come and visit our facilities, right? They're asking, how are you doing this? And you can definitely see it opens up a new world to them, that we're not in a basement, like making like weird plants or something, right? So I think that those are some of the key things that we as a bigger company have to continue to invest in, which will help with that acceptance piece and being able to bring new technologies, opening up our doors. You have a lot of transparency initiatives, but I think it is important to have companies like Pairwise do a lot, publish about their work, right? I think it's great to read about all that, and that Dan is here and you know, doing a lot of outreach and engaging all over the world, right? So it's that I think will be key to getting these technologies accepted. We can do our part as Bayer for sure, but I think it's really key to have smaller companies, universities do the same thing and reach out and be an advocate for those new technologies, which I think can really be helpful in that acceptance.
I want to make a couple comments here too. One is I think what you just said, Lieselot's really important. That hopefully with this technology, at least, there are more players, which will help. That it is more diverse in terms of the players and the applications, cetera. Of course, for that to happen, the right regulatory systems have to. The second thing is by investments like companies like Bayer and a company like ours that can lead to an actual consumer experience with the technology, nothing trumps that. So I don't think an educational deficit model will work. You cannot explain this enough. Cannot speak about it enough. Well, we tried that, in my opinion. But what's remarkable is, just as an example, I mean, I don't know how many people have eaten the Impossible Burger, but that's allowed me and my vegetarian daughter to eat a burger together. I remember talking to them early on about how they approached acceptance. Like, do they start with the mission? Do they start with explaining the GMO technology? How do they approach the acceptance piece? And the answer was, tell them to eat it. And so with us in the salads, and some of you have heard me talk about this before, but we gave away 6,000 salads that were gene edited about a year and a half ago at several street festivals, Texas, Washington, and California. We told them 3 different ways it was gene edited. And they ate it. And then we got 3,000 to respond and survey because we gave them hats. And asked what they liked, open-ended. What didn't you like? Nowhere. It was about 0.7% responded with citing the technology as a negative. About 0.6% cited the technology as positive. So that's slightly over 1%. Were the only ones who mentioned the technology because they'd eaten the salad and thought it was great. Same thing with people eating the Impossible Burgers. They don't care that it's a GMO, or now the purple tomato. So like this kind of investment and leading to another player and something that the consumers can experience, then will lead to the acceptance. And you still have to do the education and talking about the science. You still have to do it. That will not win the day. It is about creating things that connect with them and then coming in afterwards, right? And also when you couple that with the data that we have that shows the younger you go, more willing they are to accept it. And we're seeing that bear out. So.
I'm going to go over Alan's head. Sorry, Alan. No, please ask, because I know you're another player in this industry that's doing something really interesting in the peanut world. So,
thank you very much for your very enlightening presentation. I am Hortense Dodo, Webingate Gene, and yes, we are developing an allergen-free peanut using gene editing. I have some specific question probably for you, Dan, focusing on USDA regulation. If you are editing a plant but you think it is exempt, would you actually send your data to USDA? I mean, based on what you said, it's going to take a long time. So what is your approach there? If you're thinking that it's going to be exempted, and also if you think that it's not exempted.
So if we think it is exempt, what we're doing right now is still sending it into USDA.
Waiting based on.
Well, so with respect to the exemptions, they're actually turning those around really quick. Those are much simpler. There's not a lot of data involved. And all of these things can also be prospective. So you could submit an exemption now, for example, even though you may not have achieved it yet, and they will give you that, which then gives you some certainty so that when you do finish making your product, that you could then enter the market. You could give that ahead of time. And having that piece of paper, I think, is a very good thing. I actually think it serves some transparency purposes and some strategic purposes outside of your question that we could talk about if you wanted to. So our view is, if we feel confident that this is exempt, let's submit it. They're actually turning them around pretty quickly. If it's not exempt and you think you have to go through the regulatory status review, well, you have to go through the regulatory status review. That's not really a choice if you want to enter the market and it's relevant to their authority. So, which means you need to plan on how long you may be there. But as I said a moment ago, you can submit those way ahead of time. Start the clock now, which is what you see everybody else doing too, which leads to a bit of a car pile up, but you can send it in early, multiple gene targets, et cetera. So that would be my recommendation. Sorry, Lieselot. Yeah.
I was just going to add, because I had that one slide with the timelines. On the left was editing 2 to 4 weeks approximately, right, Dan? Something like that for exemptions, if it is indeed exempt. If not, go through the RSR process, which can take that lengthy timeline. So yeah, I sort of agree with what Dan said. Maybe just one thing that I think is really great that some countries have implemented too is that you can actually go and consult with them before you even have a product. You can throw out like a theoretical idea. You don't even have to show data, and you can consult with them really early. Like I think Argentina has adopted that process, a lot of countries in South America, right? So that's, something to keep in mind as well that I think is really great. And I think, good for a lot of developers to ask really early thoughts from the agency without making it formal, without anything being published, without even having a product in your hands.
Hi, Randy Dunn. Thank you all for your presentations, very interesting. If we think about the evolution of regulations for genome-edited microbes and for plants, really curious about the spectrum. Kind of on the one end, it would be complete harmonization of all regulations from a global standpoint. And on the other end, sort of complete national level regulations where there's no harmonization at all. I think there's pressures we see kind of pushing things in both directions. So where do you sort of see this end up in some years from now? Are we going to move towards maybe what we all would like is a suitable set of harmonized regulations, or is it going to stay more in the national level where it's much more difficult to navigate getting a new product approved in many different countries?
I think unfortunately for the microbes and I think the plants as well, Europe is going to do its own thing. And I think that we will not be able to have truly globalized harmonization with the regulations. I think what will end up happening, and honestly, it may be what's best is regional. So LATAM may be on one harmonization if we can get the US and Canada to be together. So that's kind of how I envision the microbes. I don't know about what you guys think in the plan.
And we actually talked about that a little bit before the presentation. What does the future look like? I would agree. I think I see it more going into a lot of different directions, unfortunately, having a lot of harmonization, which creates a lot of burden for developers because you have to know your way around. And so we talked about the case study, right? So it's a very complex landscape. But that's what I see for the near future, just because there's a lot of politics involved as well. Natasha mentioned the EU. There's tons of member states that all vote, right? And nobody's necessarily aligned. So that creates a lot of diversity. But that's not necessarily something that would stop commercialization of products, right? As we talked about earlier, there's the GABA tomato in Japan, right? There's mustard greens in the US, right? So that doesn't necessarily have to stop innovation. It just means that that technology is going to go to countries that have a science-based enabling framework in place. And those countries will reap the benefits of those products. And the other countries that don't follow suit, they won't have access to the technology, fortunately.
My goals are lower than yours, Randy.
I'm thinking long term.
Yeah. I'm not sure harmonization is the goal, right? For me, it's about risk proportionate, but of course, that's kind of balls and strikes. Like the big picture, the big game is about trade, and there's a lot of economics involved in that as well as the politics. I'm sure that China does what it does, right? And it creates certain advantages if they have a more onerous system. And so we'll see where those go. But I am encouraged, again, like places like Latin America and on gene-edited plants. Argentina and Brazil right now, we're talking about have a single window between them. Submit, and they'll both take that submission, they'll move it through their own process. That's not quite harmonization. It's not exactly reciprocity, but it's efficient. And they fundamentally have the same sort of risk proportionate approach. That to me is a really maybe achievable goal. And there's all kinds of discussion like that with respect to at least gene-edited plants. I know that you're in microbes, particularly coming out of the Southern Cone, right? So I think that that's where I'm willing to be optimistic. Yeah.
Thank you. Yeah.
So Alan, do you still have a question? Is it worthy of being the last question? Is it no pressure.
High expectation on me.
Go ahead.
Okay, I'm Alan Ayers. I'm a retired Bayer, 34 years, predecessor companies. Jack didn't let all that into his bio a while ago, but He and I are NC State grads. We were in grad school together. So, but I am senior technical advisor for Innovative Vector Control Consortium now. We work on insecticides for mosquitoes and malaria. So we have a global mission and so why am I sitting here today? Because I was in regulatory and I supported regulatory. And y'all did a great job today on this gene editing plants and especially in these vegetables. But my question is this, and I loved your slide that you said in the past years, you started in 2018. Well, we were working on biotech in the 80s, 1980s. We didn't even know what to do with it in our own plant. This guy, he came along in the 90s. We all worked together, but I mean, actually, we had a gene back then. And my question is today, with all of the experience that we've had over the last 30 to 40 years, why is there still so much unpredictability and uncertainty with USDA, EPA, and we can't push this thing on through? Because y'all gave really good examples. Why? Single deletions, the mutations we were doing way back, and we were sapping plants with deadly rays and all this kind of stuff. You know, it was like a Frankenstein back then, but it's not. And to that question too, in the my question is on with pesticides. You know, EPA does want to move to a better system. Risk 21, you know, that came out with this process, trying to quit asking for redundant studies, killing more animals, doing modeling. So why can't we develop that system in the gene editing, look at it holistically? My colleague Laura just loves that holistic word I use now.
Systems approach.
Systems approach. And then you can use something like the Risk 21 concept and say, EPA, we don't need to do that. You can see that this, we can get a waiver here and a waiver exemption. Is there any discussions moving on that, especially in biostimulants? We were talking about this, Natasha, 10 years ago. And I'm going, this is great. And they all came about because of genetically edited plants combined with biologicals. We do have a systems approach. And so, Sarsa, we all started this thing to try to discuss these items. Yeah. So I'm just interested, is there any way we can move in that direction? Are we moving in that?
Yeah, and I think the systems approach, I mean, it's a really great and future vision idea, right? I think it would be supporting, because we talked about some examples of products coming to the market. What do you do with really complex plants, right? That can maybe have hundreds of edits. Do you go for each of those edits through the USDA 100 times? Even Canada, right? You have to characterize still each of your edits. So what is a more enabling system? And I think a systems approach could be supportive to, you know, futuristic products that we might see come to the market. I know there's actually a couple of Europeans looking into this as well. In the Netherlands, there are some publications from University of Wageningen that sort of thinks through some of those concepts for on the biotech, on the GM side. So there are conversations that I think are ongoing in certain parts of the world. I haven't seen an actual, let's say, group effort on this on the industry side, but I think that's definitely something we could and should explore further again as the technology evolves, because some of these frameworks that we see in those green countries, they're now enabling the current technologies and some of the current products that we see come onto the market. But what's next? And what's that future technology? What does that look like? So can we come up with a totally different shift in paradigm also from a regulatory perspective to enable this to come to the market? Because it won't stop here with just, you know, single base pair edits. That's maybe good for now, but it doesn't support future products. So those are, you know,
Oh, go ahead, Natasha.
So, yeah, Alan, you know, we've been working on biostimulants for a while. Even, you know, you and I kind of worked together when I was in regulatory talking about it. And one of the things that we found on the microbe side, because again, it's way more developed in the plant side than it is on the microbe, the capacity building of the regulators, right? So, you know, it's companies go in, developers go in, and they don't know what they don't know. So then the fear and overregulation possibility is there. I do have hope. Again, Latin America is gangbusters, right? You can actually, I think we said numbers, you can potentially get a new product out of Latin America in 10 months compared to EPA 18 months, right? And the possibility for new technologies globally. I mean, the RNAi, right? So Europe before was a depth, a darkness for new technologies, and the possibility of RNAi, foliar RNAi being able to be registered in Europe because the application is in a country there, will open the doors for, I know, our technologies in the future. So the microbe technology. So, I mean, it's hope. But I think, again, even though consumer-wise, as Dan mentioned, education may not be the top priority, but with regulators, you do have to go in there. It has to be something where we can let them know that this technology isn't scary and help guide them. And I'm not going to say guide, help educate them on what the companies are doing. And so maybe once they get that, and they can train the other people to actually review the products correctly. So that's kind of the strategy that we're thinking with the microbes on that side.
Real quick.
Yeah. I think the scope of your question was, I mean, you were really speaking about the US and part. And I would just say that the driving force and the issue that we have in the US that has not changed is the regulators have a great fear of litigation. And so what they did is they put out rules quickly and in their mind that were going to be very helpful. That is the place that they were coming from. But because they needed to make something that they felt was highly defensible and therefore enable the technology, then they had to write them a certain way, and so that they are not as helpful as we'd like them to be. So the way to move forward on this, where USDA is going, is put something out for public comment, right? Because there is an instrument built into the rule that says that we can change this by proposing new things, which has to go. So now give us the opportunity to create the record, make it better, so you can defend it. And so when we get into some of these issues about what could be achieved through conventional breeding and therefore exempt, or shouldn't be regulated by EPA, et cetera, they have to now, they put rules out quickly again, because they felt they needed to. But now we have to have the opportunity to build the record. Of course, we could do multiple edits and polyploids, and that's a thing, and build the record so that they can say, go ahead, now we feel like we can defend it. That's the issue.
That's building more experience. Build the record.
Build the record.
Okay, I'm gonna.
We've
got to wrap it up, but you guys can carry on afterwards. But I feel bad because I said 15 and it went to 25. So apologies. I had one, but I'm going to hold that back. But I want to say thank you to three groups. So first of all, thank you to you guys for sticking around. Hopefully, you know, you really enjoyed this topic. Thank you to the four speakers. And Jack, of course, who had to leave earlier. Thanks for a great panel discussion, particularly around the regulatory. I know if there are any students or faculty in the room that have an invention pending, then you know who Kultaran is now, and please, you know, reach out to him in his office. And thirdly, I want to say a big thank you to Kathleen, again, who put this all together, but couldn't do this bit, at least today, but I know she's got some presents for the speakers. But thanks, everybody.
Yeah, I think so.